It is now my great pleasure to introduce our next speaker, Marjorie Brand. Marjorie did her PhD with Laszlo Toro at the Louis Pasteur University, identifying and characterizing the TF2C transcription complex. And for this work, she was awarded the prize for the best PhD thesis at the University of Molecular Biology. From there, she moved to Seattle to the Fred Hutch, where she conducted her postdoc work in the lab of Mark Grodin, studying long-range transcriptional control at the human beta-globin locus. She then started her lab in 2004 in Ottawa in Canada, taking a position at the University of Ottawa and the Ottawa Research Institute Hospital. As an independent scientist, she's received numerous awards and accolades, including being appointed a Canada Research Chair, being named to the Royal Society's College of New Scholars, and from the hospital being given the Correction Researcher of the Year award. In her lab, her main research interest has been the regulation of gene expression at the level of transcription and chromatin, specifically looking at hematopoietic stem cell differentiation and how that may be misregulated in leukemia. And with this, taking a multidisciplinary approach, including relative and absolute proteomics to tackle these questions. You should all definitely read or reread her papers because they're great. And I'm really excited to hear her seminar today. So with that, please join me in giving a very warm, if virtual, welcome to Marjorie Brand.
Thank you very much for the very kind introduction. So it's a great pleasure to be part of the seminar series. I was able to watch some of the previous seminars, and those are really great. So in my lab, we are interested in the transcriptional regulation of hematopoiesis and erythropoiesis. So here I have shown this hematopoietic tree of differentiation. And as you can see, hematopoiesis is a multi-step process which the hematopoietic stem cell differentiates to give rise to all the cells present in the blood. And today I will focus mostly on erythropoiesis, which leads to the formation of red blood cells, and also megakaryopoiesis that leads to the formation of platelets. Now, this process is highly regulated by a number of transcription factors and cofactors, and these proteins work together as networks to promote differentiation towards specific lineages. Now, what's interesting is that the transcription factors that drive erythroid differentiation are expressed at very high levels at the specific stage when they are important to promote differentiation. However, most of these factors are also expressed in the hematopoietic stem cell, although at very low levels, which suggests that the amount or the dosage of transcription factors plays a critical role for cell fate determination. Now, the importance of the dosage of transcription factors for cell fate decision was established early on by reprogramming experiments in cell lines. So, for example, in 1995, the lab of Thomas Graf was able to reprogram a myeloblastic cell line into a different cell types depending on the amount of the ectopically expressed transcription factor GATA1. So specifically, they showed that high levels of GATA1 will drive differentiation towards erythroid cells, while lower levels of GATA1 will drive differentiation towards the eosinophilic lineage. Now, these early experiments therefore really demonstrated that different amounts of the GATA1 protein can promote alternate cell fates. Now, it is the same thing for during native hematopoiesis, where it has been proposed that the transcription factors that drive alternate or mutually exclusive lineages are co-expressed in bipotential progenitors, and that changes in their relative levels or changes in their stoichiometry is important for the cell fate decision process. Now, a number of gene regulatory networks have been established in an attempt to model these cell fate choices that occur in these bipotential progenitors. And here I'm showing one of the examples of this network model of cell fate choice that occur in this MEP, which is megakaryocyte erythroid progenitor. So it's a bipotential progenitor that must choose between an erythroid fate and a megakaryocytic fate. Now, this hematopoietic tree of differentiation that I have shown you so far was a commonly accepted model for hematopoiesis until 5 or 6 years ago, when the use of single-cell RNA-seq transformed it into something that looks more like this. And in these new continuous models of hematopoiesis, there are no differentiation steps per se, but instead the cells are gradually transitioning along hematopoietic lineages. And in this context of continuous models of hematopoiesis, there are also no bipotential progenitors per se, but there are only progenitor cells with cell-fate probabilities that are becoming more and more restricted as the cells gradually progress along the hematopoietic trajectories. Now, in these new types of models, the exact role of transcription factors and the importance of their quantitative changes for the process of cell-fate decision remains unclear. So our goal is to establish a dynamic model of erythropoiesis that can integrate these quantitative changes in transcription factor protein levels over time. Now, our model system to study human erythropoiesis is an ex vivo differentiation protocol where the CD34 positive hematopoietic stem and progenitor cells are first isolated from cord blood or from peripheral blood or from bone marrow, and those cells are then differentiated towards the erythroid lineage by using a combination of growth factors and cytokines. Now, here you can see the morphological changes that occur during this process of erythropoiesis. And at day 0 here, you can visualize the hematopoietic stem and progenitor cells. And at the end of differentiation at day 26, you can see we obtain a complete enucleation of the cells. Now, to better characterize the process of erythropoiesis at the single cell level, we decided to use mass cytometry or CyTOF, which allows one to measure protein in single cell. Now, for the CyTOF experiments, we harvested cells at regular intervals during the course of erythropoiesis, and we barcoded them separately with palladium isotopes prior to mixing them and staining them together with a cocktail of 27 antibodies, including cell surface markers and transcription factors. And then we did clustering analysis of the 27 markers at the 14 time points, which allowed us to identify 18 different cell populations. Now, because we barcoded our sample, we could then do a temporal analysis of these cell populations during erythropoiesis, and this allowed us to reconstruct the dynamic trajectory of erythropoiesis from the early multipotent progenitors to the terminally differentiated erythroid cells. And this allowed us then to examine the expression of transcription factors in single cells within these different populations along the erythroid trajectory. So the first question we wanted to ask was whether there is co-expression of antagonist transcription factors that promote mutually exclusive cell lineages. So we looked at the expression of this factor here called FLY1, which promotes differentiation towards megakaryocytes, and this transcription factor here called KLF1, which promotes erythroid differentiation. And we looked at the expression of these two factors in this early progenitor here called the MEP, which we defined by a combination of 7 cell surface markers. And as you can see here, we found that the vast majority of those cells do co-express these antagonist transcription factors. So next, we wanted to look at how the level of these, the protein levels of these transcription factors are changing during the course of erythroid differentiation as the cells progress along the trajectory. And you can see here in single cells, and here we have aggregated the results. So we found that there is a gradual increase in protein levels of KLF1, and at the same time, a gradual decrease in protein levels of FLY1. But when the ratio between these two factors is changing, we have not observed a drastic switch after a specific population. Instead, what we see is that it does take several cell populations for this ratio to be fully resolved. And next, we induced the ectopic expression of a flag-tagged version of the non-erythroid factor FLY1 in early progenitors, and again followed differentiation by CyTOF. And when we do this time we don't see one, but instead we see 2 different trajectories. So in addition to the erythroid trajectory here, which is shown by high level of expression of the alpha globin, we also see a second trajectory here, which corresponds to the megakaryocytic trajectory, as shown by high level of the megakaryocytic marker CD41. And when we followed the cells that express FLY1 by introducing an anti-flag antibody in our CyTOF cocktail, we could see that only the cells that express FLY1, shown here in blue, are differentiating towards the megakaryocytic lineage, while the cells that do not express FLY1, shown in green, continue to differentiate along the erythroid lineage. So these experiments show that the ectopic expression of FLY1 is able to deviate the cells from their preferred erythroid trajectory to take on a megakaryocytic fate. So altogether, these results support the concept that quantitative changes in transcription factor protein levels in individual hematopoietic progenitors are key determinants of cell fate decisions during erythropoiesis. Now, one must keep in mind that CyTOF measure protein levels through antibody staining, and like all antibody-based approach, it's very good at measuring the quantitative changes in the relative level of a single protein over time or between different samples. But it does not provide any information on the relative levels between several proteins within the same sample. And therefore, we still don't know the stoichiometry between transcription factors. So why do we want to know protein stoichiometry? First of all, because we want to better understand how these changes in protein stoichiometry can regulate cell fate decisions, but also because despite everything that we have learned so far from genomic studies looking at the binding of these transcription factors in the genome, we still don't know how many transcription factors are there compared to their number of binding sites. And we don't know if the cofactors are present in limiting amount compared to the transcription factors, in which case the transcription factors would need to compete to recruit them to their binding sites, or whether these cofactors are present in large excess, which would facilitate their recruitment to specific binding sites. Now, if one is to measure stoichiometry and changes in stoichiometry between proteins, it is necessary to use approaches that provide an absolute quantification of those proteins. So for this, we decided to use a selected reaction, a targeted mass spectrometry approach, which is called selected reaction monitoring, or SRM, which when coupled with the spiking of known amounts of isotopically labeled peptides can provide an absolute quantification of proteins. Now, recently, several papers came out using quantitative mass spectrometry approach, but very few of them are using SRM. So I would just like to take a few minutes to explain why, in our case, we wanted to use this particular approach. So first, I will say that mass spectrometry is not inherently quantitative. And what it means is that the signal intensity that is measured by the mass spectrometer does not only depend on the abundance of the peptide, but also on other parameters, such as the amino acid composition or the ionization efficiency. And what it means is that it's not unusual to be in a situation like this one, where you have a very high abundant peptide, like the blue peptide, producing a low intensity mass spec signal, while a low abundance peptide like the red peptide will produce a very high intensity signal. And therefore, to be able to overcome these limitations and still being able to provide some quantitative information from mass spec data, a number of approaches have been proposed. And a very popular approach is called the proteomic ruler method, proposed by the lab of Matthias Mann. And in this method, it's possible to estimate copy number of proteins by dividing the mass spec signal by the mass spec signal produced from histones that are used as an internal standard. So this is a very good method, and it's high throughput, so you can quantify a high number of proteins in a single experiment, and it's pretty good at estimated copy numbers of proteins. So however, in our case, we wanted to be able to distinguish even subtle differences in the stoichiometry between proteins. And therefore, we decided to use a different approach, which is based on the spiking of known amounts of isotopically labeled peptides that are used as internal controls. So in our approach, every individual peptide is quantified by using an isotopically labeled version of itself, as illustrated here, which is called the SIL peptide. This method is extremely sensitive. It has a very large dynamic range for quantification, and it's currently considered as the gold standard for protein quantification by mass spectrometry. So to being able to establish SRM assay for our protein quantification, we collaborated with Jeff Ranish and Mark Gillespie from the Institute for Systems Biology in Seattle, and they have developed an optimized SRM assay for over 100 proteins. So we decided to measure different types of proteins, including DNA binding transcription factors, but also co-repressors, co-activators, some chromatin remodeling enzymes, as well as some subunits of the general transcription factor machinery. And at the end, we were able to quantify about 103 proteins at 13 different time points. I should also mention that even though it does take time to establish those SRM assays, once they have been established, they can be reused to quantify the same protein in a different cellular environment. Now, because we wanted to build gene regulatory networks, we also wanted to measure mRNA levels. And for this, again, we harvested cells at regular intervals during the course of erythroid differentiation. And we measured mRNA by RNA sequencing and proteins by SRM. So here, focusing on the RNA-based data, over time, I'm showing you this RNA-seq-based erythroid trajectory from the early hematopoietic stem and progenitor cells to the differentiated erythroid cells along these two principal components. So you can see there is a very nice progression along this trajectory. Now, if we look at the 103 transcription factors that we have quantified by hierarchical clustering, you can see that during differentiation, we see different clusters of transcription factors that appears to be temporally co-regulated. So the first question we wanted to ask was whether there is a good correlation between protein and mRNA during erythropoiesis. So first we measured the correlation between protein and mRNA as illustrated here. And we measured this correlation at the different time points during erythroid differentiation. So as you can see here, the cells that are in the process of differentiating have a pretty good correlation between mRNA and proteins. However, this correlation is extremely low in the hematopoietic stem and progenitor cells at day 0. And this is consistent with previously published data showing that the hematopoietic stem cell have an extremely low protein translation rate. So we also wanted to look at the correlation between RNA and protein changes over time, as illustrated here. And what we found is that for most of the genes, there is a positive correlation. However, for some genes, there is low correlation and sometimes even a negative correlation. So here I'm showing as an example the FLY1 and GETA2 genes, for which, as you can see, there is an almost perfect correlation between the changes in RNA levels and protein levels during erythroid differentiation. But for other genes, such as GATA1 or TAL1, you can see that the protein levels is increasing much faster than the RNA level, suggesting an important contribution of the post-transcriptional regulatory mechanisms. Now, if you're interested in looking at other transcription factors that we have measured, we have done this website here. And I think I'm actually going to skip that in the interest of time, but we found that some factors are completely uncorrelated between mRNA and proteins. And the fact that we found major discrepancies between mRNA and protein abundances for master regulator of erythropoiesis suggested to us that the gene regulatory networks should not be limited to mRNA, but should also integrate protein levels. And this is what we attempted to do in collaboration with computational biologist Ted Perkins and his postdoc, Daniel Sanchez-Saltavul. So they attempted to build a dynamic network model of erythroid commitments that can incorporate these quantitative changes in transcription factor protein levels. So in these models, the RNA are considered as the targets, the proteins are considered as the regulators, and proteins can be activators or they can be repressors. And basically, they used differential ordinary differential equations in order to be able to explain the kinetics of the changes in mRNA levels during erythroid differentiation by the changes in protein abundance that occur during this same erythroid differentiation. So here is a dynamic model of erythroid commitments that they have established around 14 transcription factors. And this is shown here at different days of differentiation. Now, what distinguishes our model from previously published model is that it's able to quantify the strength of the identified regulatory relationships. So for example, if you focus on RUNX1 and GETA2 here, you can see that the activation of GETA2 by RUNX1 is much stronger than the activation of RUNX1 by GETA2. The model is also dynamic in that it reveals changes in regulatory relationships over time. So for example, here at day 0, you can see that the model was able to capture cross-antagonisms between the SP1 and the GATA1 transcription factors. And you can see that there are differences in the intensity of these regulatory relationships. We can also see here the very strong regulatory relationships that implicate proteins that are important in stem and progenitor cells. And these regulatory relationships progressively decrease in strength, and they are fading away. And at the end of differentiation, they have completely disappeared. So as I started to mention, we are also able to correctly recapitulate the timing of the transcription factor cross-antagonisms that underlie cell fate decisions. So at day 0 in uncommitted cells, we found this cross-antagonism here between SP1 and GATA1 that regulates a very early cell fate decisions between the myeloid and the erythroid lineage. Now at day 2 of differentiation, we captured a second cross-antagonism between KLF1 and FLY1 that regulates a subsequent cell fate decision between erythroid cells and megakaryocytes. Now, another interesting aspect of our model is that it allows us to compare the strength of the different transcription factors to the regulation of their target genes. So if we focus, for example, on the activation of the GATA1 gene here, you can see that the transcription factor TAL1, its contribution is almost twice as important as the contribution of this other transcription factor GATA2 to the activation of the GATA1 gene. What's interesting is that it's not because GATA1 is a stronger activator of the gene per se, because as you can see here, in fact, TAL1 is a weaker activator of the GATA1 gene compared to GATA2. But what happens is that TAL1 is 3 times more abundant than GATA2. And therefore, what this shows is that TAL1 is a weak but abundant activator of the GATA1 gene, and GATA2, even though it is a stronger activator, overall it contributes less to the activation of the GATA1 gene because it is less abundant. So with these types of model, we are really able to quantitatively dissect these regulatory relationships between transcription factors, which is important if we want to be able to control erythropoiesis in vitro or in vivo. So everything I showed you so far was about DNA binding transcription factors. Now, as we all know, these factors work through the recruitment of cofactors, and therefore we also wanted to measure by SRM the amount of coactivators. So we started with the histone acetyltransferase P300. Now, according to the current literature, P300 has been shown to interact with dozens of transcription factors, so we expected P300 to be highly abundant in the nucleus. However, much to our surprise, we found that not only P300 is not present in excess of the transcription factors, but it is in fact present at a lower abundance than almost every single transcription factor taken individually. So then we decided to look at the other histone acetyltransferases, CBP and CAT2A or GCN5. And as you can see, those are just as rare as P300 in the nucleus. So then the next step was to look at the antagonist enzymes, the histone deacetylases. And as you can see here, those are much more abundant than the HATs. So there is a quantitative imbalance between histone deacetylase and histone acetyltransferases in the nucleus. So then we decided to look at a broader range of cofactors. And as you can see here, all the coactivators that we have measured are very low abundance, while the co-repressors are extremely highly abundant. So for example, if you focus on CHD4, which is the enzymatic subunit, one of the enzymatic subunits of the NERD co-repressor complex, you can see that there is an almost 50-fold molar excess of CHD4 compared to the coactivators. Now, this result may explain why the NERD complex has been identified as an interacting partner in almost every single proteomics screen that have been published so far. So we found that co-repressors are very highly abundant, co-activators are very rare. So where do the transcription factors fit on that scale? And the answer is that they fit somewhere in the middle. So here I have plotted the copy number of transcription factors in log scale. And you can see there is about an order of magnitude difference between coactivators and transcription factors, and another order of magnitude difference between the transcription factors and the co-repressors. Now, we were able to detect these very large differences in abundance because we looked at proteins. Because when we do the same analysis with mRNA, we found that there is no statistically significant differences in the abundance of the transcripts that code for these different classes of proteins, which suggests that these differences in abundance are regulated mostly post-transcriptionally. And this is also consistent with this result here, where we have treated the cells with cyclohexamide to inhibit protein translation. And as you can see, the co-activators appear to be much less stable than the co-repressors, which suggests that one of the reasons why the co-activators are so rare in the nucleus is because those are in fact very unstable proteins. And this is true throughout erythropoiesis. So here I have plotted the average copy numbers of these different classes of proteins during erythroid differentiation. And as you can see, the co-activators are there between 100 and 1,000 copies per nucleus, and the transcription factors between 1,000 and 10,000, and co-repressors between 10,000 and 100,000. So overall, these data suggest that the nucleus is a highly repressive environment where co-activators are limiting. And this suggests also that the DNA-bound transcription factors must compete with each other in order to be able to mediate the recruitment of this very limited number of co-activators. Now, one way through which these DNA-binding transcription factors can increase their likelihood of being able to recruit those rare co-activators is by assembling themselves onto enhancers. So we wanted to ask the question, how does the number of active enhancers and co-activator molecules compare? So we looked at the number of molecules of co-activators that have been shown to associate to active enhancers, which are the CBP/P300 histone acetyltransferases and the MLL3, MLL4, H3K4 monomethyl transferases. And to estimate the number of active enhancers in those cells, we did the ATAC-seq experiments, which we analyzed with hint attack to identify the motif for transcription factors. And then we intersected these regions with predicted enhancers from database. And this is a number of predicted active enhancers that we ended up with. And as you can see, those are in a similar order of magnitude as the number of co-activators that are present in the cell, suggesting that maybe the formation of active enhancers depend on the availability of those co-activators. So here is our model. I have plotted here the transcription factors that are highly concentrated with a very limited number of co-activators, while the rest of the nucleus is highly enriched in co-repressors. And we think that this restriction of the abundance of co-activators in a highly repressive nuclear environment may be a very important mechanism for concerted gene regulations, and it could be particularly important in multipotent progenitors to prevent high level of co-expression of genes from different lineages. So I will stop here and acknowledge people who did the work. So I have already acknowledged our collaborators. From my lab, this project was led mostly by a postdoc, Carmen Pali, with the help of two bioinformatician postdoc, Karti Sivaraman and Zinab Mokhtari. And I thank you all for listening, and I'll be happy to take any question. Thank you.
Thank you. That was a fantastic talk. I really enjoyed it. So we've got a couple questions coming in. I'll remind people you can either type your questions into the Q&A box, or you can raise your hand and I'll unmute you and you can ask your question yourself. So our first question is from Matthew Levine, who asks, how do you explain, he's talking about the first part of the talk, how do you explain the exogenous FLE1 expressing cells? How do you explain that only the exogenous FLE1 expressing cells are detected in new cells, in cells going to the new trajectory? Are the cells going in the normal trajectory, just having a lower level of the exogenous expression?
Right.
Yeah, and the normal keep the same fate.
Yeah, that's what we think. It's a question of amounts. Also, we use lentiviruses, right, to ectopically infect those cells, and the lentiviruses on primary cells do not infect all the cells. So it just happens that the one that have been infected and have expressed high enough level to go to the megakaryocyte trajectory goes there. The other cells could be just not enough or just have not been infected by the lentivirus.
Yeah, And then next is Carmelo Farai, who I'm going to unmute and allow him to talk.
Can you hear me?
Yes.
Okay, so I have two quick questions related to the fact that there is this different profile in terms of RNA production and abundance of the protein. There is any correlation in terms of this discrepancy and the amount of the final amount of protein that you can find in your differentiation? So, for instance, the proteins that somehow they need more abundant in order to be reaching the same level of activation, do they correspond to genes that are normally low expressed and they get more translated, or vice versa?
Yeah, so I think I understand the question. So we have not. So what we notice is there is a low correlation for proteins that are extremely abundant, and we think that's because a lot of it is regulated at the RNA level and also at the translational level. There is a little bit of a better correlation for proteins that are low abundance, especially when they start to be activated. But even that is not true for all of them. For example, we have GCN5, CAT2A, which is completely uncorrelated, and between RNA and protein. I mean, and this is an extremely low abundance protein. So
yeah,
I guess
let me
rule.
Let me rephrase a little bit. So I remember this graph where you were showing that there was GATA2, and I don't remember which one. It was the other protein, transcription factor, that were regulating GATA1. And somehow, despite the fact that GATA2 was lower in terms of abundance, the level by which he was able to transactivate GATA1, it was higher. And somehow this you explain by the fact that, yes, there is less GATA2, but it attacks more and eventually is more effective than the other protein that compensate by the amount. So the question is, do you see any kind of pattern in the sense that the genes that are having discrepancy between RNA and protein abundance can be somehow making a specific kind of condition in this regulation?
Yeah, we have not found any pattern like that would be able to explain that. So by pattern, you mean specific DNA binding sites, or that they would all go up?
What you show is these two phenomena, right? Let's say two behaviors. One is low abundant protein, but highly affinity or highly transactivated. And the other one is abundant protein, but kind of less efficient. And I wonder if the one or the second categories belong more to the genes that are having a perfect correlation RNA protein or divergency RNA protein production.
Yeah, no, not really. Like we could not find anything like that, but we haven't really looked at it. So it's possible, but I just don't know.
Okay, thank you.
All right, and then we've got another question from Zhaoli Wu, who asks, if D repressors are at excess, you would imagine a competition between co-repressors and co-activators for transcription factors. Can you speculate or hypothesize, in this case, how do active transcription complexes form?
Oh, I see. So yeah, so that would be on the enhancers, because you have all the activators that comes together and form a high affinity, right, platform to be able to recruit them. Now you could, in our model, you could have the same one of these transcription factor, but not being together with, like being isolated, and another gene, for example, would not have a high affinity enough to be able to recruit the co-activators, and it would be able to recruit a co-repressor, which also could explain that the same transcription factor can actually be an activator or a repressor, depending on the gene, and that has been observed, I think, by a number of experiments.
Yeah, thank you. Okay, so I think we'll. If you have more questions, we'll call it there for the seminar part. So thank you again, Marjorie, for the fantastic talk and discussion.